my humble opinion, the single most important biochemical reaction, especially to us, is cellular respiration. And the reason why I feel so strongly about that is because this is how we derive energy from what we eat or from our fuel, or if we want to be specific, from glucose. At the end of the day, most of what we eat, or these carbohydrates, end up as glucose. In future videos, I'll talk about how we derive energy from fats or protein. But cellular respiration, let's go from glucose to energy and some other byproducts. And to be a little bit more specific about it, let me write the chemical reaction right here. So the chemical formula for glucose, you're going to have 6 carbons, 12 hydrogens, and 6 oxygens. So that's your glucose right there. So if you had 1 mole of glucose, that's your glucose right there. And then to that 1 mole of glucose, if you had 6 moles of molecular oxygen running around the cell. And this is kind of a gross simplification for cellular respiration. I think we're going to appreciate over the course of the next few videos that one can get as involved into this mechanism as possible. I think it's nice to get the big picture. But if you give me some glucose, if you have 1 mole of glucose and 6 moles of oxygen, through the process of cellular respiration, and so I'm just writing in kind of a big black box right now. Let me pick a nice color. So this is cellular respiration. You'll see it's quite involved, but I guess anything can be if you want to be particular enough about it. Through cellular respiration, we're going to produce 6 moles of carbon dioxide, 6 moles of water, and this is the key, this is the super important part, and we're going to produce energy. We're going to produce energy, and this is the energy that we're going to, that can be used to do useful work, to heat our bodies, to provide electrical impulses in our brains, whatever energy, especially human bodies, but it's not just humans, it's provided by this cellular respiration mechanism. And when you say energy, you might say, hey Sal, in the last video, did you just, well, if you missed out on the last video you watched, you probably saw that I said that ATP is the energy currency, for biological systems. And so you might say, hey, well, looks like glucose is the energy currency for biological systems. And to some degree, both answers would be correct, but to just see how it kind of fits together is that the process of cellular respiration, it does produce energy directly, but that energy is used to produce ATP. So if I were to break down this energy portion of cellular respiration right there, some of it would just be heat. It just warms up the cell. And then some of it is used, and this is what the textbooks will tell you. The textbooks will say it produces 38 ATPs that can be more readily used by cells to contract muscles or to generate nerve impulses or do whatever else, or grow or divide or whatever else the cell might need. So really cellular respiration, if it produces energy, it's a little disingenuous. It's really the process of taking glucose and producing ATPs with maybe heat as a byproduct, but it's probably nice to have that heat around. We need to be reasonably warm in order for our cells to operate correctly. So the whole point is really to go from glucose, from 1 mole of glucose, and the textbooks will tell you, to 38 ATPs. And the reality is this is in kind of the ideal circumstances that will produce 38 ATPs. I was reading up about it a little bit before doing this video. And the reality is, depending on the efficiency of the cell that's performing cellular respiration, it'll probably be more on the order of 29 to 30 ATPs, but there's huge variation here, and people are really still studying this idea. But this is all cellular respiration is. In the next few videos, we're going to break it down into its kind of constituent parts, and I'm going to introduce them to you right now, just so you kind of realize that these are parts of cellular respiration. The first stage is called glycolysis. Glycolysis, which literally means breaking up glucose. And just so you know, this part, the gly for glucose, or the glyco for glucose, really. The glyco for glucose. And then lysis means to break up. When you saw hydrolysis, it means using water to break up a molecule. Glycolysis means we're going to be breaking up glucose. And in case you care about things like word origins, glucose comes from the gluc part of glucose comes from Greek for sweet, and glucose is indeed sweet. And then all sugars, we put this "-ose" ending, so that just means sugar. So you might think it's kind of a redundant statement to say sweet sugar, but there are some sugars that aren't sweet. For example, lactose. Milk, it might be a little bit, but when you actually digest lactose, then you can turn it into an actual sweet sugar. But it doesn't taste sweet like glucose or fructose would actually, or sucrose would taste. But anyway, that's an aside. But the first step of cellular respiration is glycolysis, breaking up of glucose. And what it does is it breaks up the glucose from a 6-carbon molecule. So it literally takes it from a 6-carbon molecule. Drop like this. The 6-carbon molecule that looks like this. And it's actually a cycle. Let me show you what glucose actually looks like. This is glucose right here. And notice you have 1, 2, 3, 4, 5, 6 carbons. I got this off of Wikipedia. Just look up glucose and you can see this diagram if you want to kind of see the details. Or you can see I have 6 carbons, 6 oxygens. That's 1, 2, 3, 4, 5, 6. And then all of these little small blue things are my hydrogens. So that's what glucose actually looks like. But the process of glycolysis, you're essentially just taking, I'm writing it out as a kind of a string. You can imagine it as a chain, and it has oxygen and hydrogen added to it, to each of these carbons. But it has a carbon backbone, and it breaks that carbon backbone into two. That's what glycolysis does, right there. So you kind of lyse the glucose and each of these things. And I haven't drawn all the other stuff that's added onto that. You can draw all bonds and other things with oxygens and hydrogens, whatever. But each of these 3 carbon backbone molecules are called pyruvate. We'll go into a lot more detail on that. But by glycolysis, it by itself generates, it needs 2 ATPs. So it needs 2 ATPs, and it generates 4 ATPs. So net, on a net basis, it generates 2, let me write this in a different color. It generates 2 net ATPs. So that's the first stage. And this can occur completely in the absence of oxygen. I'll do a whole video on glycolysis in the future. Then these byproducts, they get re-engineered a little bit, and then they enter into what's called the Krebs cycle. They enter what's called the Krebs cycle, which generates another 2 ATP. And then, and this is kind of the interesting point, there's another process that you can kind of say happens after the Krebs cycle, but we're in a cell and everything's bumping into everything all of the time. It's normally kind of viewed to be after glycolysis in the Krebs cycle. And this requires, let me make sense, this requires oxygen. It requires oxygen. So let me be clear, glycolysis, this first step, no oxygen required, or doesn't need oxygen. Oxygen, it can occur with oxygen or without it. Oxygen not needed. Oxygen not needed. Or you could say this is called an anaerobic process. This is the anaerobic part of the respiration. I'll write that down too. Anaerobic. Maybe I'll write it down here. Glycolysis, since it doesn't need oxygen, we can say it's anaerobic. You might be familiar with the idea of aerobic exercise. The whole idea of aerobic exercise is to make you breathe hard because you need a lot of oxygen to do aerobic exercises. So anaerobic means you don't need oxygen. Aerobic needs oxygen. Anaerobic means the opposite. You don't need oxygen. So glycolysis is anaerobic, and it produces 2 ATPs net. And then you go to the Krebs cycle. There's a little bit of setup involved here, and we'll do the detail of that in the future. So then you move over to the Krebs cycle, which is aerobic. It is aerobic. It requires oxygen to be around. And then this produces 2 ATPs. And then this is the part that frankly when I first learned it confused me a lot, but let's write it in order the way it's traditionally wrote. Then you have something called, use the same colors too much. You have something called the electron transport chain. Electron transport chain. And this part gets credit for producing the bulk of the ATPs, 34 ATPs. And this is also anaerobic. It requires oxygen. So you can see, if you had no oxygen, if the cells weren't getting enough oxygen, you can produce a little bit of energy, but if you don't, but it's nowhere near as much as you can produce once you have the oxygen. And actually, when you start running out of oxygen, this can't proceed forward. So what happens is some of these byproducts of glycolysis, instead of going into the Krebs cycle in the electron transport chain where they need oxygen, instead they go through a side process called fermentation. Fermentation. For some organisms, this process of fermentation takes your byproduct of glycolysis and literally produces alcohol. That's where alcohol comes from. That's called alcohol fermentation. And we as human beings, I guess fortunately or unfortunately, our muscles do not directly produce alcohol. They produce lactic acid. So we do lactic acid fermentation. Write that down. Lactic acid. That's humans and probably other mammals. Humans. But other things like yeast will do alcohol fermentation. Alcohol fermentation. So this is when you don't have oxygen. It's actually this lactic acid. And if I were to, sprint really hard and not be able to get enough oxygen, then my muscles start to ache because this lactic acid starts to build up. But that's just a side thing. If we have oxygen, we can move to the Krebs cycle, get our 2 ATPs, and then go on to the electron transport chain and produce 34 ATPs, which is really the bulk of what happens in respiration. Now, I kind of said this as an aside, and, to some degree this isn't fair, because while these guys are operating, they're also producing these other molecules, or they're not producing them entirely, but what they're doing is they're taking, I know it gets complicated here, but I think over the course of the next few videos we'll get an intuition for it. In these two parts of the reaction, glycolysis and the Krebs cycle, we're constantly taking NAD, or NAD+, and we're adding hydrogens to it to form NADH. And this actually happens for 1 molecule of glucose, this happens to 10 NADs, or 10 NAD pluses to become NADHs. And those are actually what drive the electron transport chain. And I'll talk a lot more about it, and kind of how that happens, and why is energy being derived, and how is this an oxidative reaction, and all of that. What's getting oxidized, what's being reduced. But I just wanted to give due credit. These guys aren't just producing 2 ATPs in each of these stages. They're also producing, actually combined, 10 NADHs, which are then used, which each produce 3 ATPs in an ideal situation, the electron transport chain. And they're also doing this other molecule, FAD, which is very similar, but they're producing FADH. Now, I know all of this is very complicated. I'll make videos on this in the future. But the important thing to remember is cellular respiration, all it is taking the glucose and kind of repackaging the energy in glucose and repackaging it in the form of, your textbooks will tell you, 38 ATPs in the exam, that's a good number, right? It tends to, in reality, be a smaller number. It's also going to produce heat, actually most of it is going to be heat. But 38 ATPs, and it does it through 3 stages. The first stage is glycolysis, where you're just literally splitting the glucose into 2. You're generating some ATPs, but the more important thing is you're generating some NADHs that are going to be used later in the electron transport chain. Then those byproducts are split even more in the Krebs cycle, directly producing 2 ATPs, but that produces a lot more NADHs, and all of those NADHs are used in the electron transport chain to produce the bulk of your energy currency, or your 34.
Yeah. Okay, so that cellular level stuff is really deep. let's simplify it up. What causes you to be hungry? The blood sugar drop. Okay? And so that stimulates you to get hungry, right? And so when you say the blood sugar drops, what do you mean? Where's it dropping at?
Those levels drop to the point where your body thinks it's sufficient enough to.
Okay, so your cells, right? Your cells are saying, I need some extra glucose. Why are your cells saying that?
So it doesn't have to
Because it needs glucose to do what?
metabolize. It's great.
For cellular respiration to create energy. And we need energy to do what?
Everything.
Everything, right? Okay. So what happens in the meantime between the time your body says I'm hungry and you take in some food? Because we can't just, oh, I'm hungry, I'm going to go eat right now sometimes, right?
Find an alternate source.
Finds an alternate source, okay? And initially that alternate source is stored glycogen in your liver, right? So your brain says, okay, cells called me up and said, I need some extra sugar because I need to create some more energy to do some more work. So the brain sends a signal to the liver and says, release some stored glycogen. And how do we get stored glycogen? So stored glycogen is the sugar that we take in that we don't use. They store it in the liver. Why? So that we can, just in case. So our liver dumps glycogen. It goes through glycolysis, which then puts it into a form of glucose that can be used by the cells in addition to oxygen to create energy. Everybody with me? Okay. What does there also have to be enough of for the cell to be able to use the sugar? Insulin. And what's insulin do?
It's basically just a drug factor.
Yeah.
There's a lot of things, but for sugar, it pushes the cell.
Yeah, so it opens the door and lets the sugar go in. Right? Okay. So let's say that we let the sugar in, so there's sugar in there, there's oxygen in there. It does its magic through the Krebs cycle and the electron chain analysis, and it creates energy. There's two waste products of cellular metabolism.
Carbon dioxide.
Carbon dioxide.
And then lactic acid is very anaerobic.
Yep. So the other acid is pyruvate acid. And that's pyruvate acid is usually not a big deal. Okay? But let's say that we're a diabetic and we don't have enough insulin and our body's screaming for sugar and we dump some glycogen, we twist it through glycolysis to glucose, it gets to the cell, it knocks on the door of the cell, there's no insulin to open the door, so what happens to that sugar?
Continues to rise.
It stays in the blood, so our blood sugar level goes up, but meanwhile the cell is still starving, so it sends a signal back to the brain says, okay, eat more, dump some more glycogen, twist it to glucose, gets to the cell, knocks on the door, no insulin, can't get in, sugar's building up, cells are still starving, so at some point the cell goes, screw it, I got to do something else. So then it uses fats, muscles to create the energy. And the waste product of cellular metabolism using something other than oxygen and glucose is lactic acid, right? And then lactic acid causes a big spiral effect of stuff, including more vasodilation, decrease in all kinds of stuff. And this is where we get the big spiral to acidosis. Right? Everybody with me? Okay. So, we'll go through a couple of slides and then we'll look at another video. So, shock sepsis and MODS, which is multiple organ dysfunction syndrome, they're very, very complex, but because they affect every single system of the body. Okay? And can we, as a human, fight off this? Can we fight shock?
Compensate.
Okay, right, so we can compensate for a while, right? Until the big vicious cycle gets to the point that the body says, I'm done. And then we go over the hill to decompensated shock. And if we stay in decompensated shock for a while, what will we end up in?
Yeah.
Irreversible shock and then ultimately death. Exactly. Right? So when you think about this, if we have decreased perfusion to the tissues or to the cells, we're decreasing the amount of oxygen and sugar that's getting to the cells to start with. Right? So they're going to dysfunction already. If a bunch of cells dysfunction, tissue dysfunctions. If a bunch of tissue dysfunctions, an organ dysfunctions. If a bunch of organs dysfunction, we have multiple organ dysfunction syndrome and shock. Exactly. So it begins with the cellular level local tissue damage, and then it can progress. All right, so we're going to skip all this because he went through all that. So aerobic metabolism uses something else. So aerobic metabolism uses glucose, amino acids, oxygen, and the ADP to produce energy, carbon dioxide, water, and heat. He kind of went through that. Anaerobic has to get it from something else, right? And the end result is in anaerobic metabolism, we're not producing as much energy. So if we're not producing as much energy and we're not perfusing any of that stuff to the end organs or the tissues, it's all going to start to dysfunction. Okay, essentially. All right, so what is microcirculation?
Small circulation.
Okay, small circulation. When I say small circulation, what do you think? Arteries, veins, capillaries. Capillaries. All right. So capillaries are what? It's where the venous side of circulation and the arterial side of circulation come together. Right? And they're so small, they only allow one red blood cell at a time to go through there. And why is that? Why do I care if only one goes through at a time?
That's a safe precaution for the body.
Yeah, it pretty much guarantees good stuff on, bad stuff off. Right? Where if I sent like 12 things through at the same time, some of it would happen and some of it wouldn't happen. Does that make sense? So that's what we're doing. This is called the crash site. That's the crutch of where all the nutrition, oxygen, and all that stuff has got to get to for us to have cellular respirations. So, homeostasis, what is that? The body tries to keep itself in a state of equilibrium or balance, right? And that includes what? Oxygen demand and supply. So if you're running and your muscles are using up energy, your body needs to create more energy. To create more energy, you have to have more sugar and more oxygen. So what does your respiratory system do?
So breathe faster.
Breathe faster and deeper. That has two purposes. Bring in more oxygen, get rid of more CO2. CO2 is acid, right? What does your heart rate do? It goes up. What does your contractility do? It goes up. Why?
Trying to feed the cells.
Feed the cells, right? Pump more blood. What's your blood pressure do? goes up. All of these things are part of the compensatory mechanism to keep you alive, right? There's a few things when we get into shock. There's 3 different potential mechanisms to cause that not to happen. If something's wrong with your pump, you have a hole in your container, or your container's too big, and you have a loss of contents, right? One of those 3 things, or a combination of those 3 things, can end up causing a shock, right? Here's a good one. When oxygen-bound hemoglobin reaches the capillaries, what does it have to do? It has to get rid of the oxygen to the cells, right? And to the tissues. So that's a huge deal. So back from paramedic school, there was this totally foreign concept that we learned. It's called the oxygen hemoglobin dissociation curve, right? And we talk about left shifts and right shifts and all this kind of stuff that we don't remember. So I'm going to show you a little video real quick of kind of an easier way to do that. Remember that.
Hello, in this video we're going to focus on lung physiology, focusing on gases and pH. We'll also look at oxygen dissociation curve. So air enters the respiratory system in the upper airways, proceeds to the conducting airways, and then onto the respiratory airways, branches of the bronchi, bronchioles, terminal bronchioles, and finally ends in the alveoli. You can think of the alveolus, which is singular, of alveoli, as the building blocks of the lungs, where gas exchange occurs, gas exchange occurring between the lungs and the bloodstream. The gas exchange in this case is between oxygen and carbon dioxide, or CO2. So just recapping the basic physiology, the heart will pump deoxygenated blood from the right ventricle towards the lungs. Deoxygenated blood means blood containing a higher concentration of CO2 with lower amounts of oxygen. In each alveolus, the carbon dioxide will be dropped off and exhaled out of the body, so removing carbon dioxide from the body. And then oxygen will be inhaled and will then reoxygenate the blood. This newly oxygenated blood, or reoxygenated blood, will then go back to the left side of the heart. The heart will then pump this oxygenated blood around the body to the other organs, such as the brain. The oxygenated blood delivers oxygen to organs, tissues. The organ tissues then uses up this oxygen to help create energy in the form of ATP. Now, as a process of creating this energy, ATP, the organs, the tissues will form a byproduct, which is carbon dioxide. Carbon dioxide is then made and is actually expelled into the bloodstream as exchange, you could say. Now, because carbon dioxide is in the bloodstream, the blood is now deoxygenated once again, and this deoxygenated blood will return to the right side of the heart. The right side of the heart will continue the cycle. It will pump blood to the lungs to reoxygenate the blood. Oxygen itself travels in the body mainly in red blood cells. Oxygen binds to these molecules called hemoglobin, and we have millions of these in each red blood cell. In deoxygenated blood, with deoxygenated hemoglobin, or deoxyhemoglobin, the structure of hemoglobin here is referred to as a tight structure. Hemoglobin, just recapping, is a protein made up of 4 subunits, each of which contain a heme moiety attached to a globin chain. And each globin chain contains iron, where oxygen normally binds to. Each of the 4 iron atoms in hemoglobin can reversibly bind one oxygen molecule.
So
Let's
you
see what
have
happens
4
when
receptor
deoxyhemoglobin
sites on each
receives
molecule of
oxygen.
hemoglobin to accept oxygen.
Well, the reaction is rapid, requiring less than 0.01 seconds. Remember, in deoxyhemoglobin, the globin units are tightly bound in a tense configuration, in a T configuration, which means that it actually reduces the affinity of the molecule for oxygen. When oxygen is first bound, the bonds holding the globin units are released, producing a relaxed configuration, or an R configuration, which exposes more oxygen binding sites. So to put it simply, you can imagine it like a cascade. Firstly, the hemoglobin is in a tense configuration with no oxygen. And then when oxygen starts to bind, it will become more relaxed, a relaxed configuration, which actually increases the affinity for oxygen even more. Kind of weird. This whole thing is called positive cooperativity. So by the end, the hemoglobin molecule is an oxyhemoglobin because it is filled with oxygen and it is in a relaxed form. So from the lungs, you now have heaps of oxyhemoglobins, which means your blood is reoxygenated really. And this reoxygenated blood is ready to be delivered to the body tissues. It's a good idea now to briefly talk about partial pressure of oxygen and carbon dioxide in deoxygenated blood and also in oxygenated blood, because it will be relevant to what we will talk about later. But you can imagine that when blood is oxygenated or reoxygenated from the lungs, it would have a higher partial pressure of oxygen, so higher amounts of oxygen, 100. And carbon dioxide is low, 40. The oxygenated blood I'm talking about are found in your arteries, such as your aorta. Deoxygenated blood, on the other hand, is very different. When we talk about deoxygenated blood, we're talking about the veins. So oxygen, it can be up to 40 millimeters of mercury, and carbon dioxide about 45. The changes in partial pressure is important to understand because it helps explain the hemoglobin dissociation curve. And we will talk about that now. The hemoglobin dissociation curve, or the oxygen hemoglobin dissociation curve, relates a percentage saturation of the oxygen carrying power of hemoglobin to the partial pressure of oxygen. Now that might sound crazy, but let's just try to understand it together.
So we're
in the
comparing the
x-axis, you have the partial
PaO2 in the
pressure of
arterial
oxygen,
blood
25
to the
100
saturation.
millimeters of mercury. And on the y-axis, you have the saturation of hemoglobin, which what we're looking at specifically is oxyhemoglobin in percentage. This saturation of oxyhemoglobin means how much oxygen is bound to the hemoglobin basically. The more oxygen bound to hemoglobin, the higher the oxyhemoglobin, and therefore the higher the saturation. So the oxygen hemoglobin dissociation curve has a characteristic sigmoid shape due to the tense and relaxed configuration interconversion. There is a plateau here where the partial pressure of oxygen is nearly high. The plateau signifies that high partial pressure of oxygen does not cause large changes in oxygen saturation of hemoglobin. So at the plateau, you can see that the partial pressure of oxygen of 100 already has an oxygen saturation of 98%. So there's not much change. Whereas before the plateau, so a partial pressure of oxygen of 50, let's just say, already has an oxygen saturation of 85%. So a dramatic change. So note that small changes at low partial oxygen pressure leads to large changes in oxygen saturation. Another example is that a low partial pressure of oxygen, such as 26, has already oxygen saturation of 50%. The reason for this sigmoid shape and why low partial pressure of oxygen has a quick increase in oxygen saturation is because of positive cooperativity. When the first heme in the hemoglobin molecule receives oxygen, the affinity increases for the second, and then the third, and so on. Now let's look at factors which can shift this curve, which are essentially factors affecting the affinity of hemoglobin for oxygen.
This is the important part.
Three important conditions affect the oxygen-hemoglobin dissociation curve. These are the pH, the temperature, and the concentration of 2,3-diphosphoglycerate, or DPG. Here is the curve normally. We will look at examples of what shifts the curve to the right and what shifts the curve to the left. When the curve shifts to the right, it means that there is reduced hemoglobin oxygen affinity. And this occurs in tissues. So basically, the hemoglobin affinity for oxygen is decreased. Remember the normal curve, the partial pressure of oxygen of 50 had an oxygen saturation of about 83 to 85%. In this new curve, the partial pressure of oxygen of 50 has an oxygen saturation of 70%. So reduced oxygen saturation. And this is because there is reduced affinity for oxygen. Things that cause it to happen include an increase in carbon dioxide, which really means a decrease in pH. Other things that causes the shift to happen include an increase in temperature and an increase in DPG or DPG. It's good to remember that the reduced hemoglobin oxygen affinity occurs in tissues in general. Because when you think about it, you want to give your oxygen to the body tissues so that they can use it. These tissues include the placenta because you want to offload oxygen to the fetus, and muscles, cells, especially during exercise. Now the curve can also shift to the left here in red. What happens here is that we see higher hemoglobin oxygen affinity. And for this example, you can think of the lungs, because it is in the lungs where oxygen comes and binds to hemoglobin. And so you would expect to see higher hemoglobin oxygen affinity here. Later, when the blood travels to the body tissues, such as the muscles, the affinity reduces, as we have learned. The other factors which contribute to the high hemoglobin oxygen affinity, so this red curve, are reduced carbon dioxide, which means higher pH, reduced temperature, and also reduced DPG, which is essentially opposite to the curve when it shifts to the right. So let's compare now. In this red curve, so a high hemoglobin oxygen affinity, if the person has a partial pressure of oxygen of 50, the oxygen saturation is already 90%. So in this video, we talked about how oxygen binds onto hemoglobin from a deoxygenated state in a tense configuration into a relaxed R configuration. And we also talked about the oxygen dissociation curve and factors which cause the curve to shift to the right, as well as factors which cause the curve to shift to the left. Hope it made sense. Thank you for watching.
Okay, so think through the oxygen hemoglobin dissociation curve. And if it shifts to the right, it allows more oxygen to get off, correct? Is that correct? Okay, so think about this. What things cause it to go to the right? Okay, 2,3 DPG. Increased temp, so something like fever or just heat generation from exercise. And CO2, right? The CO2 is acid. So is there going to be a place where we shift from a right shift to a left shift. So if we are in shock, if we're in deep shock, right, and we're trying to get rid of CO2, and we blow off all our CO2, and that causes our pH to go up, it shifts the curve to the left. We also get cold, shifts the curve to the left, right? We get low perfusion, shifts the curve to the left. And now I got all this oxygen that can't get off. Now what happens to my tissues? They start getting mad, right? And then when they get mad and they ain't got no oxygen, then they start creating lactic acid, and then you get this big circle spiral downward of your patient getting sicker on you. Any kind of shock. Any kind of shock. Yeah. So, that kind of makes sense? So what I would tell you about this stuff is you kind of need to know the 2 or 3 things that make it shift one way or the other. Okay? All right, so 4 stages of shock. In the initial stage, the microcirculatory beds decreases. So what does that mean? So you have pre and post capillary sphincters. What's a sphincter? A valve, right? It's a valve. And so the first thing that happens is both of those sphincters close. And what's that doing? It's trapping blood, right? Okay, is that a good thing or a bad thing? It's a bad thing. Why is it a bad thing? Can't get rid of CO2 and you can't get oxygen in, right? And what else is it going to end up doing? It's going to end up getting acidotic, right? But that's the first thing that happens. So that blood gets hypoxic, right? It says that the body uses physiological mechanisms to maintain cellular homeostasis. What are we talking about? Our heart rate increases, contractility increases, respiratory rate and depth increases. What do our vessels do? They constrict to do what? Increase blood pressure, right? How does the body know how to do that? Because we have pressure sensors in our carotid arteries and our aortic arch that sense drops in blood pressure, sends the signal to the brain, who then sends it to the respiratory center, vasomotor center, and cardiac center to initiate this compensatory mechanism. What's driving this? Which part of the nervous system is driving this? Sympathetic nervous system, right? And what's the primary thing that drives the sympathetic nervous system? Epi and norepi, right? Adrenaline. That's your fight or flight, right? Would you say that the fight or flight is what helps you compensate for shock? Absolutely. Okay? All right. So, we start seeing a decrease in cardiac output, the decrease in blood pressure, sympathetic nervous system takes over. It causes vasodilation in the skeletal muscles, coronary arteries. Your sweat glands start to produce diaphoresis. Your heart rate increases. Your lungs, respiratory rate and depth increases, and your pupils dilate. Right? Why are we sweating?
Your body's starting to work hard and create heat, so your body needs to stay cool.
Okay, that's one part of it, and the second part of it is what's another, other than dissipating heat, what's another mechanism of sweat?
Sodium.
To get rid of stuff, right? Specifically getting rid of toxins. Exactly, getting rid of toxins. Okay? We get vasoconstriction in our skin, renal, and gut. Okay? What does arterial vasoconstriction do to preload? It makes it go up, correct? To compensate. how would we know that preload is going up? What diagnostic tool do we have that we could look at and go, I think they're compensating? Maybe, but what if they're on a beta block? What is a diagnostic measurement of preload? What?
Cap refill.
Diastolic blood pressures.
Oh.
Yeah, your diastolic blood pressure starts going up. Your systolic blood pressure stays about the same, right? Your diastolic blood pressure is going up because your body's trying to increase preload to help increase output. Make sense? So what do we get? A narrowing pulse pressure, right? As a general rule, your systolic stays about where it's at or goes up a little bit, but your diastolic comes up pretty significantly to try to increase preload. Now, how does that happen? It happens a couple ways. One, it happens with arterial vasoconstriction, and the other way it happens is muscle contractions pushing that blood back into the system. So if I push the blood back into the system, fill up the container more, and then I vasoconstrict to increase the pressure, I get more back to my heart. Make sense? And the way it does that is through peripheral and gut vasoconstriction. So what does this do? This shunts the blood to more important things. Your gut doesn't need it, and your skin doesn't need it. Your heart, your brain does need it, right? What else? Why else am I constricting the vessels to my renal system? To conserve what? Salt, right? Because what follows salt? Water. So I want dump some antidiuretic hormone in the system. I want to decrease the perfusion to my kidneys so I don't filter as much water and pee it off, because if I pee it off, I pee off salt. Water follows salt. Now we got this big vicious cycle that's going to end up making our container empty. Does that make sense? All right. So, when we decrease renal blood flow, we increase the renin-aldosterone system, which helps us retain sodium and water. We release the ADH, which again helps us retain sodium and water. We also release cortisol and adrenocorticotropic hormones from the anterior pituitary gland, and then our adrenal medulla, which is where? Sits where? What? Sits on top of your kidney, right?
The adrenal, yeah.
Yeah, your adrenal gland. Yep. And that's where we produce our epi and norepi. All of these things together increase sugar levels. And why are we increasing sugar levels? Because we need more energy, right? To fight this thing off, right? What's going to happen eventually when we run out of glucose stores or glycogen stores? We're going to end up with no sugar, right? We're going to end up hypoglycemic. If we end up hypoglycemic, we're going to end up using fat and muscle to create energy, and we're going to end up with acidosis and all this different bad stuff, right? So where would this have a major effect? Think about a kid. Does a kid have a lot of major glycogen stores? No, because they don't have much body fat to them, right? So let's think about a kid who is already hypermetabolic, right? Let's think about why are children's respiratory rates higher than adults? Huh?
Metabolism is higher.
Their metabolism's higher, which causes them to create more CO2, which causes them to breathe faster to get rid of it to maintain their pH balance, right? Does that make sense? Okay, now think this through. If they don't have any glycogen storage, and we're not giving them any glucose, they're going to run out quicker. Right? So if you think about a burn kid, we not only give them fluid resuscitation for the burn, we give them maintenance dose fluids of glucose to prevent that from happening. Right? And you will find also, even like in an adult burn, they're hypermetabolic. And so when we eat a 2,000 calorie meal a day, when you're trying to fight burns, we're giving them 4,000, 5,000, 6,000 calories a day because their metabolism is so sped up to try to fix the burns that they can get hypoglycemic super quick that way. All right, so we get decreased cardiac output, decreased blood flow to the lungs, increased physiological dead space. So what does that do to our oxygen level? What is physiological dead space to start with? This doesn't get to the alveoli, so nothing happens, right? There's several things that can increase alveolar dead space. One is blood flow. So think about this. Blood has to go by the alveoli to get oxygen on and CO2 off, right? So if I had a big pulmonary embolism, I'm not getting blood by the alveoli, so I'm getting air to it, but I'm not exchanging gases. So that air is low in oxygen, right? Okay? If I have a big chunk, let's say I have one lobe that's not getting any blood flow to it, that's one lobe full of air that's not getting oxygen. So what's it doing to the rest of the oxygen or the rest of the air in that lung? It's diluting it, right? Does that make sense? I'm diluting it. That's what dead space is. It's air that's it's not getting to the alveoli, or it's getting to the alveoli, but it's not exchanging gases. You can increase dead space with stuff like pneumonia, pulmonary edema, pneumothoraxes, hemothoraxes, pulmonary embolisms. Can pulmonary embolism cause shock? What kind of shock? Obstructive shock, right? So, this increase in physiological dead space causes a decrease in our O2 levels, which then stimulates our chemoreceptors to tell us to breathe faster and deeper, right? Because we're trying to increase the PO2 and decrease the CO2, but instead we're getting an increase in CO2. Why? It's the same reason. We're not exchanging the gases. So there's CO2 in there, but we're not getting the oxygen in there. So our CO2 is building up. Our oxygen's going down. Okay? We start breathing faster because of the sympathetic response. This causes what? Respiratory alkalosis. And what does CO2 do to your vessels? CO2 causes dilation, right? So if I blow CO2 off, I cause constriction. If I cause constriction, what do I do to blood flow? Decrease it. If I decrease blood flow, what do I do to oxygen delivery? Decrease it. If I decrease oxygen delivery and sugar delivery, I cause anaerobic metabolism, which creates acid. And it's a big vicious cycle. Does that make sense? Kind of how that works? What is the most sensitive organ to hypoxia? The brain, right? So anytime an organ is deprived of its nutrients, sugar, oxygen, energy, it starts to dysfunction, right? Sometimes we can see that clinically. Sometimes we have to see that with like lab values. Okay? But obviously we can see a decrease in level of consciousness. Right? All right. So what happens when our body's compensatory mechanisms can't keep up anymore? We go into decompensated shock. So here's what happens. Our goal is to aggressively treat compensated shock so that it doesn't decompensate. When we go into decompensated shock, there's two things that you should think about to help you figure out if that's happening. One is they get hypotensive, right? And hypotensive is a pressure below 90 systolic or 30 points lower than their normal. So if they run at 150 over 100 all the time, they could be hypotensive at 110 because their body, especially their brain, is used to that high blood pressure. Okay? Most people, you're going to use a systolic of 90 as your threshold. And I want you to use level of consciousness. Because if they're at 110 over something and their LOC is altered, what does that tell you? They have diminished oxygen delivery to their brain, right? So it doesn't matter what their blood pressure is at that point. So when I say altered mental status, give me some examples. Huh?
Like conditions.
Now, stuff you would see that would lead you to believe they're altered. Confusion. What else? Anxious and restlessness are the first ones. Confusion. What about this? Head bobbing, right? So any of those things should be considered in your head as progression of compensated to decompensated shock. Here's what happens next. Remember those capillaries that we closed off on both ends? Now those sphincters relax. Okay, and what did we say was in those capillaries? Stagnant blood, right? So specifically what's in there? Acid. So now, let's say all the pre and post capillary sphincters in my legs let go. Now I have 2 or 3 liters of acid now in my systemic circulation. And what's that going to do? My body's going to go, what the hell? Because it's not going to like that acid, right? pH is going to drop big time. That's going to move my curve where? Moves to the right. Okay? So that should mean I'm going to put more oxygen at the cellular level. But what's the problem? My perfusion sucks. So it can't get there to get off. That's the problem. Okay? It requires early aggressive fluid resuscitation and vasopressor support if we're going to live through it. When we get to the point that we can't help them compensate anymore, we're going to go into irreversible. And where we get irreversible is multi-system organ failure. So their kidneys fail, and then that causes their liver to fail, and then that causes something else to fail, and then they end up in ARDS and DIC and all kinds of weird stuff, right? And it's almost 100% fatal. So irreversible shock is that decompensated shock that lasts so long that we get end organ damage or end organ failure out of it. So, all of these things are progressive, right? So some of the things that we can get from shock, if our heart's ischemic and cold, what's it going to do? It's either not going to work right or it's going to throw off some extra stuff, right? PVCs runs a VTAC because what? The heart has that property called automaticity where it can create its own impulses. Usually when it decides to create its own impulses is when it's ischemic and cold and doesn't like it. So it throws off PVCs, runs a VTAC, stuff like that. Low blood pressure to the lungs causes pulmonary hypertension. Pulmonary hypertension is hypertension in your pulmonary artery. Okay? The significance of that is what's pumping into my pulmonary artery? My right heart. My right heart has to have something in it to pump to it. Yeah, if I have decreased preload, it's trying harder to pump to my lungs. My lungs are going, I need some extra blood. I'm going to clamp down. Heart's having to work harder to get it in there. And again, big vicious cycle. How come we get pulmonary edema when we get into shock? What does acid cause your cellular membranes to do? Leak. Okay? Where are they leaking? Into the tissues, right? When we're talking about the capillary beds around the alveoli, that's going to cause it to leak into the alveoli. And now what? Can water exchange gases? No. So now what? We're getting more hypoxic, more hypercapnic. And again, hypercapnic causes vasodilation. Vasodilation does what to your blood pressure? And it's already low. Yeah, a big vicious cycle if we don't fix it. Other stuff down here, you can get stress ulcers, ischemic bowel with release of toxins, hypoglycemia, on down the road there. So, there's two types of MODS. Primary MODS is a result of direct insult, such as trauma. Secondary MODS is more progressive to the organs. So it's more common because typically what's causing MODS in the secondary? Shock. Right? Shock is the more common reason we have MODS. Early affected organs, brain, kidney, liver, adrenal glands, and heart. Now, why is that important that the adrenal glands stay perfused?
Keep pumping out adrenaline so you can compensate.
Right. Okay, so think about this. What drugs can we use for pre-medication or sedation for RSI? Ketamine, fentanyl, atropine, etomidate. Etomidate. So, have you ever heard the term catecholamine dependent? What does that mean, Colton?
Not for sure, to be honest with you.
Okay, so a catecholamine dependent patient means that the only reason they have a blood pressure is because of catecholamines, epi and norepi. Okay, causing you to have vasoconstriction. Right? That's my daughter. Causing you to have vasoconstriction. Correct? Okay. Y'all can go in there,
Okay.
and I'll be in there in a minute.
Okay, come here first. Carolyn.
So, if you're catecholamine dependent, and those catecholamines are coming from your adrenal medulla on top of your kidneys, and they're also, well, that's the easiest way to look at it. Now, in septic shock, they are catecholamine dependent. What's a side effect or negative effect of etomidate? It turns off your adrenal cortex. So if I turn off your adrenal cortex, what are you not going to produce anymore? Epi and norepi. But you were dependent on those, so now what's going to happen to you?
Blood pressure and everything's gonna change.
Right. So, etomidate is contraindicated in sepsis for that reason. That's why you need a different med for that. Right? It's very, very more pronounced in kids than it is adults. All right. Why don't we take a quick break, and then we'll get into the different classifications of shock.